summer. One man, one decision, one voiceover. Critics are calling it sensational. Earth as we know it will never be the same.
Researchers are in race to create coronavirus vaccine. Researchers racing to produce a vaccine for the Wuhan coronavirus believe they may have taken significant steps towards finding an effective method of immunization. At least 3 pharmaceutical companies, as well as teams of researchers around the world, have been working on different approaches to producing an effective vaccine. These efforts accelerated after Chinese academics publicly posted the genetic sequence for the virus. A research team at the National Institutes of Health in Maryland said they had prepared a modified version of a key section of the virus to encourage the body to produce antibodies against the disease. As well as the NIH, the Coalition for Epidemic Preparedness Innovations has announced it would commit $11 million to 3 programs led by the companies Inovio Pharmaceuticals and Moderna, and the University of Queensland, with the hope of having a viable vaccine in production within 16 weeks, although testing for safety and efficacy will take much longer. With different teams competing to produce a trial vaccine, it remains unclear who is leading the race. Kiznaikia Corbett, lead researcher on the NIH coronavirus vaccine team, said her group had been concentrating on the spike proteins, the spear point of the virus. This built on work done during the severe acute respiratory syndrome outbreak that first emerged in China in 2001, another coronavirus thought to be similar to the Wuhan virus. The virus looks like a knobbly ball covered in protrusions that widen at the tip. These are spike proteins, which are able to dock to a receptor found in the lung membrane known as ACE2, which allows the virus to enter the body. Corbett said that coronaviruses are generally just a large family of pathogens of which now 7 are infectious to humans. SARS and the Wuhan coronavirus are similar across 82% of the entire genome. But from a vaccine point of view, we were interested only in the spike, which is 70%. SARS and MERS have shown we could drop some stabilizing mutations into the backbone of the spike after a colleague did this with the S protein of the respiratory syncytial virus. Since 2017 we have been looking at how you give the body the right kind of messenger RNA that will attract the right type of cells to produce the right type of antibodies to produce the protein vaccine. We've been doing these sequences for the last 3 years now, where we can basically plug and play, and are now able to look at any coronavirus spike sequence. One of the problems, said Corbett, is that the coronavirus spike protein is the largest of its kind to infect humans. One of its kind to infect humans. One of the reasons it's so bad to look at is because it's so big and floppy, she said. The NIH team is among several that have been working on solutions to the wider problem of coronavirus since the first emergence of SARS. Many of the teams noted that vaccine work would be further advanced if not for the loss of interest in research after the SARS outbreak was brought under control.
Thanks for watching Global Movies, your global entertainment. If you like it, please subscribe our channel for latest news and movies.